All right, unmuted. Did I do it again? I did it again. Can you hear me? If you can hear me, thumbs up. If you can't hear me, thumbs down. If you can't hear me, I don't know how you know that it's thumbs down though. Cool. So I have a little bit of a cough today, so I'm trying to do the socially responsible thing since there's somebody else in the room with me. But it's so much easier to talk like this, so I'm going to take it off. And if you don't understand why I would wear a mask while I'm sick and that other person will wear a mask while they're not sick, you should watch our microbe video on Jump website. It's kind of fun. You grow microbes on pieces of cheese. I enjoyed it. So it's nice. I see people that I've seen before. That's really cool. Sometimes I don't remember if I saw you in a real at Jump Steam or if I saw you during the on-demand ones or the live ones, but it's good to see familiar faces, which is always cool. If you don't remember me or if you've never met me before, I'm Shannon Egley. I'm the anatomical coordinator at Jump. Jump is a collaboration between the University of Illinois College of Medicine and OSF Hospital right next door. So we are here for simulation training in medicine. We help med students get through and learn their skills with actually hand-on practice. And when they're doctors, then they come back and get more hand-on practice so that when you're seeing a physician that's at OSF or actually any of the areas that use us, that you're getting somebody that's done the procedure a lot of times and they're really good at what they do. So it's just like you with practicing. So this is my lab here. If you've been here, awesome. Come back as soon as you can. We do a lot of STEM in class, like dissecting hearts or inflating lungs, where we have STEM students in. We also have the on-demand things like dissecting sharks, and we send hearts, lungs, brains, eyeballs, oh man, what else? Pieces of bone, all those different things to your house so you can dissect too. So if you haven't done that, you should look it up. That's pretty fun. I figured since we can't bring you here, we try and send the lab as close as we could to your house. So this is where we do practice surgeries. You can see the surgical table over that shoulder. Oh, that shoulder. You can see actually a crypt table over here, a morgue table. You can see we have the surgical lights and everything so that we can imitate the surgeries. When you come in for a STEM, we try and use all of the equipment so you get a chance to see, is medicine really what you want to do? Is it the direction you are interested in going? Maybe you're curious after you come in and work with us. And hopefully doing these STEM programs like this, you'll get just as curious. And I've had people that ask me, why am I doing sea creatures when I'm not a veterinarian? And I said, because they're all animals. A lot of the systems that work in us as humans work in these other animals. It doesn't matter if it's lamprey or a shark or a pig or anything, we can compare. And once you learn how to work on one animal, a lot of the principles actually apply to humans. So hopefully get your curiosity going. Let's see, what else do I do? I'm clinical associate of anatomy at the University of Illinois College of Medicine. So I teach the med students anatomy before they become doctors. And then what's cool is that once they become doctors, they can come here in this lab and practice their procedures in this lab too. So it's fun watching them grow and develop. It's like watching students, I guess, in high school or college or anything grow and actually use their skills. And I've seen some of you before. It's fun when I bump into you again and you remember the things that you learned in some other class. So hopefully you also remember that I love the actually moment. So listen for them, things that are interesting that you can use outside of science. They're kind of fun, weird, kooky, but people say something strange in conversation, you go, actually, and you just give them that finger and say, actually, hold on a second, let me explain that in a nice, polite way. Never lost any friends because of my giving them the finger, but you never know. Okay, so we don't have as much time as we usually have to do dissection, so I really want to kind of jump straight into it. The first thing you should do is there's a chat down here or up there. I can't remember as a viewer which way it is. But if you turn on your chat, that way if you have a question, if you let me know, I keep glancing up at the chat box and I'll try and catch it as soon as possible. I've muted you, kept you on mute just because feedback gets really nasty and weird. But if there's a question you have, just throw it in the chat. I will also be doing 2 screens. So I'll have a screen projecting down for the dissection. You can go right along with me, but I'll also have the screens because I do a lot of animated things with my head and face and hands and stuff. So there'll be those 2 screens that pop up. First, let's get your tools out. So let me flip on that extra screen. There it goes. You don't have this. Of course, this is my placemat to help me know where the center of the screen is. It's also important because it shows how OSF and University of Illinois actually had a kind of a brain baby jump simulation. So we work with those 2 as collaborators, and PNC also funds this to help us get the sets to you inexpensive so that you can do more of these at a pretty inexpensive cost. You're going to need this and this. We'll put this down for now. We're just going to go through this set right now. So as we open it, here are the things you'll need. You need your pad, obviously. Your pad should have 6 pins in it up here. If you only have 5, that's okay. If you have 6, that's perfect. You may not use them all at one time. But the one trick with it is anytime you take a pin out, put it back in or always know where it's at. Because if you pull this out, you stick it into one of the creatures, it gets the formalin on it, and then you stick your finger with it. It's itchy and irritates. It's not poison to kill you, but it just stinks. It's all day. So always know where your sharps are. Second thing would be another sharp. All that sharp, but this is the probe. One side's pointy all the way around. The other side's and pointy. I always keep that in my mat too, so that I know where it is. I hate putting something down and putting a over the top of it and going to grab the specimen and getting poked with a sharp. It's better just to know where your sharps are. And the third sharp are your scissors. And they look like scissors, but if you drag your finger along them, they'll cut you, so it's always best to keep them closed and keep them somewhere safe. Next, your PPE, personal protective equipment. It's a term that most, because of COVID, so you'll want your goggles, 2 gloves, and an apron. So we don't want to splash anywhere. The apron's to protect your clothes if this stuff gets on you. I've been wearing gloves, and this is the 3rd time I've done this. I already smell fish through my gloves. Ugh. So you don't want this to splash directly on your skin or splash on your clothes. So that's what the apron's for. Because it's not going to hurt you, but it's stinky and it's like cutting an onion. The smell just stays with you and stays with you. I was drinking my coffee and I was like, what is that fishy smell? Ugh, it was my hand, even though I washed my hands. Woo, bonus pack, I got 2 pens. You should have 1. Pickups, forceps, tweezers, whatever you want to call them. Oops. A ruler, a bookmark that has a website you can go to and get an app that has a bunch of games and stuff you can play. They're pretty cool. It also has like cartoony things that are 3 or 4 minutes long about the circulatory system, digestive system, nervous system, etc. to help give you an overview of the different systems focused on human. And then the last piece, last 2 pieces, I got a dirty book, but you should have the book that says Comparative Anatomy of Mysterious Sea Monsters. And we're comparing the anatomy of the sea creatures, but we're also comparing it back to human. So like I said earlier when a friend asked me about why am I doing what a veterinarian would do, it's because we're all pretty much the same on the inside. We're going to talk about the systems, which ones have a circulatory system, GI tract, nervous system. We'll compare to the ones that don't, etc. And then you have a garbage bag, and the garbage bag is your tablecloth. So moving along fast, but I'm going to keep watching to make sure that you're keeping up. Oh, and you have a whole bag of critters. Yeah. So what you want to do is whatever space you're going to work on, pull all of your stuff out of the way. And you're gonna take the bag. You're not gonna open it like a garbage bag. It is a garbage bag, but you're not gonna open it like that. You're gonna find the end that has the seam. So you have one side that has a perforated edge, the other side has a perforated edge, but only one has a seam. That would be the bottom of the garbage bag. Grab that side, open it up. It's like a bed sheet or a tablecloth. You pop it open like this. And cover your cable so that wherever you're working is right in the center of your garbage bag. So you have a splash zone that's about 5 feet across, about 3.5 feet wide. So hopefully you don't splash that much. And then you'll put all of your stuff right on top. That way when we're done, we're actually gonna put everything right back here in the same place and then wrap it up in the garbage bag and throw it away. So you don't have any more cleanup. The table will be clean when you're done and everything. Okay, let's PPE up. First thing you need, gloves. If the gloves are a little tight, that's perfect. I like tighter gloves because it lets me use my senses. If I have tight gloves, I can feel all the ridges. If I'm squeezing something, I can feel the texture of it. Like when we work with the shark, I can run my fingers along the shark and actually feel all the scales on the shark. So if they seem like they're tight, good. Gloves on. If you break a glove, you can always grab a Ziploc bag or like a grocery bag from Kroger or Walmart, whatever, and put it over your hand. You can still feel through it. It's really thin material. That way you don't get your hands as stinky. Next, your apron. I have a lab coat. I don't need an apron, but you just pull it out, open it up. There's a loop that goes around your neck. Somewhere in there. Here. A loop that goes around your neck, and on the other side are straps that go around your waist. So don't let it dangle, because if it dangles, sometimes it drifts to the side, and if you splash your clothes, then yuck, it stinks. I'll let you put that on if you haven't already. You can't see everybody. It's like a lobster bib. I'm wondering if you'll actually like the idea of lobster after this, or if you'll detest the idea of ever eating lobster again after you cut open a crab that's been preserved. And then your glasses, goggles. I have my own goggles, my own glasses, I shouldn't say goggles. They can put on. Hopefully it touches your cheek so that if you splash something up, it doesn't go under the goggles or under the glasses. If you have small glasses, you can always pop these on over your glasses. All righty. Next book. Comparative Anatomy, Sea Monsters. So this is a lot of fun. When I taught college biology, we did some of these dissections, and it's fun going back and doing them again. So think like a scientist. This is actually in every, oops, this is not the right book. It is the right book, but it's a proof. So there might be notes. After this one. It's used, but still a goodie. So I open it up. That's how I knew it's a different color. So think like a scientist. And I always say this, if you've been around me before, think like a scientist. Because you learn so much more and you're actually more useful to us in the world. I don't know a better way to say that, but when you think like a scientist, you question things, you make observations. I don't know if you do this with your friends, but you know certain patterns with things. You learn about your friends by watching them and how they do things. If you're somebody in sports, by watching the patterns of the players that play against you, you know how to predict what they're going to do. I mean, you're a scientist. It doesn't matter if you're an athlete, but if you observe, you question, you make an estimated guess of what they're going to do, suddenly you're a scientist at the same time. When you were a baby, you took like a yellow ball or a purple ball or whatever, and you put the ball in your mouth. You're feeling it with your fingers. You feel it with your lips, since that's lips and fingers are the most sensitive parts of your body. You're trying to understand. You're learning what this round ball is. You're observing this round ball. You're questioning, what is this? What do I know about it? You're touching it to your face. You smell it. Now, whatever you do with it, you drop it and watch it bounce. And then you develop this hypothesis. Well, if it's yellow and it's round, it must be a ball, and it does these certain things. But then, after you've taken that information and you repeat and modify, when you find something that's kind of round and kind of bumpy, and it's a fruit, and you bring it close to your mouth, and you can smell like that citrusiness, and then you put it in your mouth, and it's bitter, super sour. Then you have to rebuild this. Now you have to start questioning yourself. Is it a lemon or is it a ball? So babies are like the best scientists ever. They are the dumbest creatures on the planet. And I have my own babies, but they are just dumb, real dumb. But they learn really, really well because they are naturally a scientist. They question everything. They try and experiment. They push their limits. I have a 3-year-old that's actually pushing his limits right now. I'm saying right now because I know at home he's doing it, seeing what he can get away with. But that's it. My dog is a scientist. I built an electric invisible fence around my yard. She got the collar. She tested the fence. She'd go around the different parts of the yard and tested to see where she could get out. She figured out if she goes close enough to the fence and stops, the little collar stops beeping and she can cruise on through. Dogs. I see the dog behind you. It's going, oh yeah, that's brilliant. So have the right tools, and that's not about the toolkit that you got. It's about what's between your ears. So make sure that when you're doing things, explore, question. Doesn't matter if it's at school. I was actually just talking to somebody else about math. If you don't get it one way, you experiment. You try different ways. You try to find the best method that works for you, and you do it. You find success and do it over and over again. It doesn't matter what you try. Keep exploring. When you're done with this kit, I only have 2 hours to spend with you, so we're going to do kind of general dissections on all of the animals, but afterwards you're welcome, if your parents are okay with it, you're welcome to dissect all you want. You can look through and find the brain. I don't think we actually look for the brain in anything. So that's something you can explore. Find the spinal cord, follow it up and look for the brain. How big is the brain in a lamprey? How big is the brain in a squid? How big is the brain in a crab? You can figure those things out. Getting prepared, we're already prepared. As long as you're thinking and you have your personal protective equipment on, you're all set. Here's the tool set. Don't jump ahead, because what I'll do is I'll explain it to you once, I'll show you once, and I'll let you do it. And like I said, I really want to just get the stuff open, so let's open that time. So comparative anatomy, you will have 5 creatures. One of them is not in the bag. One of them looks like that. You all got that, right? Sweet. So originally, scientists thought these were plants, but we actually found after studying that they're animals. So I thought that'd be great to put in. They can look like this. They don't all look the same. They can be darker in color, but it's a sponge. And you can test how many people know this. Does anybody know what a sponge is while it's alive? What's the name for a sponge while it's alive? It's a sponge. Yeah, so I told someone I was putting sponges in the kit, bath sponges, and they're like, oh, it's an animal? What was it when it was alive? And I said, it was a sponge. And she goes, but when it was alive, what did they call it? And I said, oh, this is technically a bath sponge when it's alive. And when it's dead, we use it as a bath sponge. So it's kind of funny. It's one of those things that a lot of people don't realize that a sponge is an animal, and they don't realize that it's called a sponge. Same name. You'll have to test that. All right, I'm going to walk through opening this up, and then I'll have you do it. You have 2 bags. You have the outer bag, and you have the inner bag. You'll want to lift the inner bag up a little bit. Oh, see, there's one in every bunch. The outer bag should be clean. Mine has a leak. So you'll leave everything in the outer bag if you have a leak. You walk over to the trash can. And oops, I need scissors. You can dump this in the sink if you let warm water run for about 3 to 5 minutes, because once this formalin is used in the tissue, it neutralizes it, so it's not as toxic to the environment. But you can drain this into the sink, let the water run if your parents are okay with it. Otherwise, just dump it into a garbage bag that you know it doesn't have a hole. So I'm going to pull this off. And there's more puddle right there. I don't know if you can see that, but there's more liquid here. I'm just going to cut the corner and drain it out into the trash. I didn't quite get the corner. There you go. I'm letting it drain. Okay. And I'll bring it back to the table. That's what you want to do. So I will give you a couple minutes so that you can go drain the bag and bring it back. And as soon as you get back, we'll start opening up the parts. Excuse me. Oops, wait. Are you dissecting over carpet in your living room? I'm pointing at you. Pointing at all of them. That's brave. But what's interesting, actually, formalin and formaldehyde that's in the preservative of this is all over your house. When carpet's made, formaldehyde's in it so that it doesn't mold or get fungus growing inside of it. Couch cushions, the fabric on the couch, paint in the walls, everything in your house really has formaldehyde in it. Otherwise, when you walk outside and you get fungus all over your shoes, like in the woods or whatever, and you walk in onto the carpet, that mold should grow on the carpet, but formaldehyde keeps it from molding, and that's why we use it as a preservative. Did you know that? It's everywhere. Yeah, inside air is toxic for that because it's the like classic formaldehyde. I'm fidgety. I feel like I should do that. What's that dance where they do this thing? I don't even know. All right, it looks like everybody's back. Cool. So if you can keep your book kind of clean somewhere, I would say do it. I'm going to cut whatever end you cut the initial cut in. Let's go ahead and cut that across. Pull your critters out. Nice. Oh, that's 2 in a row. My crab had busted legs. Nuts. Oh well, luck of the draw. I'm gonna hang this here so it doesn't rip all over everything. This still freaks me out. Just that mouth. It's like a leech, except the leeches are they insects? They're a hemolymph. It's a type of worm. Is a worm in the insect group? I'll have to look that up. I'll have to look it up. Google. Google knows everything. Squid, crab, there's one leg. Sea star, most people call it starfish. I call it a starfish. And sponge. 1, 2, 3, 4, 5. All there. Excuse me. Perfect. So you open all of your specimens. Nice. First thing you want to do is kind of look at the different animals and compare in your head. What do they have in common? What do they have different? I mean, some obvious things. There's a shell. This has a shell-like skin. I don't know how to compare sponge. These two have smooth, slimy skin. This one has an eyeball. That one has an eyeball. This has eyeballs we'll find later. I mean, there are things they have in common. Things that are obviously different. And it's mostly the physical characteristics that we define species or different types of animals in. What they have in common, put them in a group. Things that are very different, but in a totally different group. So as we go through these, here's some questions you want to ask. So observe and compare in your head. What do these things have in common with the last animal that I talked about? What do they have in common with human? Suggest a function. Why do you think they have that? Why do you think that these arms on the squid are the way they are? Why do you think the tentacles are different? Why do you think that the mantle, or this big body part, is shaped like that with those fins on the top and its head's way down here? That seems so weird. I mean, even an octopus is more like that. Why is a starfish so spiky on the top? Why are there 8 legs on the crab? And then you want to also think back to the things that we define as life, which we're going to go over on the next page. So we'll define life, we'll describe it, we'll say what's living, what's non-living, and then since we already know these things are all living, what do they have in common that qualifies them as living? Right? Terms, if you've done STEM with me before, you know I always start with anatomical directions. So Eastern and Western, one of those days. Eastern and Western medicine are 2 very different types of medicine. Western medicine is based off of like European medicine from hundreds of years ago. Eastern medicine is based off of China and like Thailand and Taiwan and Japan and all of that. They believe a little bit differently. They have their own language. Western medicine has its own language. And we use these terms based off of Latin because it's an old language that we base like Spanish and French and Italian and all those languages off of. So it's good to actually get familiar with the terms, especially if you're going into any kind of biology, because you use these over and over again. So when we talk about something's back, we're referring to dorsal. So if I'm talking about the lamprey and I'm talking about a fin on its back, there's its face, there's its back, I would call this the dorsal fin, right? I can say something towards the front or towards the back would either be towards the head, I refer to it as cranial, because we think of a skull as a cranium, or anterior. So we call this fin on the back, we call it the anterior dorsal fin because it's the front one. This one we call the posterior dorsal fin because posterior is saying towards the back, which we also call caudal. Caudal is Latin for tail. When we say something's proximal, we mean it's in close proximity. It's close to something. So if I talk about the nasal hole right here, the little hole that you can see bubbles coming out of, is proximal to the eye, I mean it's close to the eye. If I say that the 7th gill slit is distal from the eye, I mean it's very far away from the eye. It's the furthest one from the eye. So it's distant, distal. If I say something is superficial, I mean it's close to the surface. If I say they say beauty is superficial, it is. It's on the surface of your skin, what you look like. Deep, personality is deep. It's what's inside of you. So those are the next 2 terms. Superficial is towards the skin. Deep is down towards the bones or the center. The last 2 terms, no, lateral means to the side. So in a lamprey, it has a right side and a left side. So lateral will be over to this side. I can say that it has an eye laterally on the right and the left. And then medial means close to the middle, like that little hole, the nasal there is medial in the center. Medial to the eyes, actually. So those are some of the terms. And I'll usually use those terms, and then I remember that you haven't had anatomy before usually, so I'll try and re-explain them each time, just so you get used to it. So what is life? Life is really summed up by biology books. There are 2 things that determine life. Does the thing metabolize? So does it use some kind of product to make energy? That's metabolism. Does it store it? Does it make it? The second thing is its ability to make copies of itself. Can it replicate? That's life. 2 sentences. Anything that can't metabolize is not alive. Right now we're dealing with the whole COVID situation, and anybody know what that is? Is it a bacteria? Is it a fungus or a virus? It's a virus. Viruses are not alive because viruses don't metabolize. They have no way to take and create energy or store energy. What they do is they invade your cell that is alive, and they use your cell for movement. They use your cell to get food. They use your cell to digest, your cell to get energy from respiration, and your cell for circulation. They're not alive at all. So if somebody sneezes out a virus, it's not like the virus is swimming through the air towards you. It's propelled like a cannonball. Once it lands, it's done. It stays there. You have to hope it doesn't land on some part of your body that it can actually get into one of your cells. And then the ability to replicate. So over here on this side, when we talk about species, these are kind of the exceptions. It's hard to define some animals as species, even though they're a real animal, because they can't replicate. A mule, I don't know if you knew this, but a mule is actually a baby of a donkey and a horse. A mule can't have babies of its own. So a donkey and a horse make a mule, and that's the end of the line for the mule. Once they're done, there's no replication. A liger is the same way. He'll love this if you've ever seen Napoleon Dynamite. That was hilarious. He talks about ligers. It's a lion and a tiger that have a baby. They make a liger, but that liger can't have babies, so it can't replicate either. I wish this would quit focusing in and out for you. Or even just in the same species, two dogs. I don't even want to try and figure that out, how a Chihuahua can actually have an offspring with a Great Dane. They're both the same species, but it's really difficult for them to replicate. So based on these rules, sometimes it's hard to define life, so we have to play it kind of fast and loose. But a virus is not alive. Diversity describes the variety in all of life. So when we talk about something like a mouse or a rat, they're actually totally different species. They're not the same. They look the same. Physically, you could describe them the same. But the way that they process food, the way that they live, the environments they live in are very different. And so we have to take that into consideration. It's like birds. If you ever heard of the Galapagos Islands, if you've taken a biology class, in fact, there's so many types of finches. And they're all finches, but they're all very different. The way that they crack nuts or what environment they like to live in, super different. So we define these different groups by what they have in common, whether it's biological functions like how they metabolize, or morphological, which means their shape. We can define it that way. So if you were looking at something like a cat, a fox, a dog, and a bear, and had to describe which ones were most closely related, the way we typically define this, what do you think would be the most closely related? Just pick two. I think it would be a cat and fox, cat and dog, cat and bear, fox, dog, fox, bear, or dog, bear. It's actually these two. So even though a fox physically looks a lot like a cat, and one of the things it can do is it actually has retractable claws like a cat, which is unique, that a dog doesn't have, all of its other characteristics make it just like a canine or a dog or a wolf. And bear is very far. It's more like a dog than a cat or a fox. But we look at all those characteristics. What makes it the same? How does it process food? How does it get food? How does it protect itself? How does it attack other things? And then we actually break it down into domains, kingdoms, phylums, class, order, family, genus, species. And what's really weird about this is you could take this and put a dog all the way through, all the way down to canine familiaris is the last part of a dog. And that's a subspecies because a species actually includes dogs and wolves. So we don't usually keep wolves as pets. So they actually came up with a special definition for a wolf compared to a dog. Again, we play fast and loose with the rules, but we try and categorize things that have features in common. And that's what we're going to do today with these, all marine animals, which means saltwater animals. Some of them can go into freshwater, which is different than marine. We're going to see what they have in common and then find out what they have different. So how can they be related and how are they different? And also to us. So I'll actually skip over the next page because really what you're looking for here, there are three main domains in all of life, millions of different species, and they all fall into three different domains. Basically a plant, a eukaryote, and a, archaea, which is like slime molds and weird things. So we're just focusing on animals, not plants, not the slime molds. And we're going to see what these animals have different. Oh, sorry. Here are a few things you want to keep in mind. When you look at it, the first thing we're going to do is, can we say that they have specialized cells? So if I look at starfish, does it have a heart that's specialized for pumping blood? Does it have a digestive system? Does it have a nervous system? If I look at a sponge, does it have the same thing? Does a squid have a heart? Does it have digestive? Does it have nervous? The same thing with the crab. So can we say that the tissues are specialized like an organ to do something? Put those in groups. Next, I guess I'm read straight down. Does the animal develop radial symmetry, bilateral, or asymmetry? Symmetry just means organization of the shape. So its body. Radial means there's one central location and everything grows out from that location. So you have a center, a very clear center of it, and everything grows in the same pattern all the way out. Bilateral means I could cut it in half, and it's a mirror image. The left side is exactly the opposite of the right side. Humans are bilateral. If I cut me in half, My left and rights are mirror images. This one, when you take a biology class, if you haven't already taken one, this is so in depth. It says, how does their gut develop? It just means, do they, this is weird to say, but when they're developing, does their anus develop first or their mouth? And does it always stay the same? So some creatures, their mouth and their anus are the exact same thing. Ugh. We're not going to talk about that much. It's gross. And then the last one, does growth occur by molting or by constantly growing? Like us, we don't shed a skeleton on the outside. We actually have a skeleton that grows from within and just keeps adapting. Other creatures aren't like that. So one of these creatures actually molts, gets rid of their shell, and grows a new one. All right, let's do some dissecting. So the first thing we're going to dissect is the phylum Porifera, and it tells you exactly what it is. It's very porous. So that would be this guy. And it doesn't matter if it looks like that or this. You can see where the base was, where it was growing. Same with this one where they clipped it off. It's nice and flat. So I added this because I just think a sponge is really weird. Originally, scientists thought it was a plant because it lived with a seaweed on the bottom of the ocean. It didn't go anywhere. It wasn't really mobile. But when they started studying it, they realized that when you have a baby sponge, it looks more like this. It has little flagella. It actually swims like a propeller. It goes to where it wants to grow, plants itself, and then it becomes sessile, which means it stays in place and doesn't go anywhere. So it starts growing up and out. It doesn't really move around after that, but it still can move. If you ever get a chance and you Google, I think it's sponge dye circulation or sponge dye pumping or something like that, DYE, like dye. Scuba diver goes down by a sponge, pours some glowy yellow dye around the sponge and waits a minute. He touches the sponge and then he watches the sponge pulls the dye in and squirts it out the top. So it doesn't even look like the sponge is moving, but it's actually moving water. It's creating current and flow. Because the inside of the sponge, all of its walls actually have these little cells with flagella on them. So they can create a current and they can actually push the water through. And they do that because they want to catch their prey. So movement, initially they move. After that, it's movement for currents, water. They filter feed. So as they're pushing the water through, bacteria in the water, I'm trying to think of other things like plankton, that's the word I'm looking for. Babies of other animals, like a blue crab has a little teeny tiny baby, it'll flow in here. The sponge catches it and the filter it, turns it into baby crab soup, and then pours it all around the sponge so that it can absorb it in. A sponge doesn't have a heart, it doesn't have a brain, it doesn't have GI tract, no circulation, but all those work together to actually create this super complex organism, nutrients and transport things. It's pretty. And then what did we do? We cut it off and rub our back and our face with it in the bathtub and paint walls with it. But I liked this picture. I thought the shape that you actually have is a leuconoid. It doesn't have a core cavity. It's just like that everywhere. So I thought, oh, let's find that actual shape. And I found a shape that looked exactly like it, and flow. How flow will come into a larger pore set and go once to get filtered, fed. And this is actually a slice of human lung. This isn't even a sponge. But your lungs are exactly the same. We bring air in through these bronchioles down into these lungs, and we capture oxygen, and then we push it back out. Same concept. It pulls with nutrients in, pushes it down, and then we'll push it back out while it feeds. Kind of an amazing creature. It's alive. Well, we killed it, I guess. It's alive all the time. How they reproduce, two ways. So here's out if you have a boy or see this side, this is totally a boy. Side totally a girl. It's all boy or girl. So the whole sponge and a girl. This sponge reproduces with itself and spit out those little flips to swim away. If I put two sponges near each other, they can swap reproductive structures and they can make a new sponge that has half the genes of each, just like parents do, like people do. half dad, half mom. But otherwise, this sponge can make its own offspring. They swim around and start populating with exactly the same genes, the same DNA. So it'll be a sponge that has all the same structure as this. Or it can break a piece off called budding. It floats in the water and plants, and it can grow a new organism. So some of these organisms can actually do that. Like you probably heard of a lizard. If you break its tail off, it grows back. If you break a piece off, it'll just regrow. Starfish, same thing. Crab, same thing. I want that. It's like Wolverine when he gets something cut off and he grows it back. I don't even know why I use that reference. I'm not even a like X-Men fan. I did get told I look like what's the Jedi, Liam Neeson. Yeah, it's crazy. And I looked at him, I was like, yeah, he's got the same big nose as me. We could be kin. So now I'm a Star Wars fan. Okay, so that's one phylum. This is like the most basic animal out there. It doesn't have tissues. It just has a couple types of cells that can convert to whatever it needs. So it can be a superficial cell that creates the shape on the outside. It can be an internal cell with the flagella that propels water. It can actually be digestive cells in between those two layers that help move nutrients or digest those baby crabs and stuff. So it's crazy. One of the impacts of ecology from the sponge is that, well, number one is they clean up. They're filter feeders. They clean the bottom of the ocean. But as far as we use it for medicine, there are studies that are looking at that inner layer of cells and the way that they can become anything. If you have somebody that has really bad burns or they have part of their skin ripped off and we put a graft on it, we're studying how we can take those cells that can mutate and become whatever cell we need it to be to actually help regrow that tissue and help glue it into place safely. So it's kind of cool because there are immune cells in there, there are digestive cells, there's every cell inside of there. There's no organized tissues. It's weird. Next one, phylum, Echinodermata. Echino means spiny or spiky. Derma means what? I know you know this. What's a dermatologist study? Your skin. So, yep. So Echinodermata means that they have a spiky or spiny skin. Which of these animals would be that? Pretend you didn't see the little spikes on this guy because those are barnacles. There it is. So we're going to need to do some cutting. I'll move the animals out of the way. So we call this a star. That totally looks like the SpongeBob dude, doesn't it? Can't remember his character's name. SpongeBob and Patrick. Yeah. So yeah, this is totally a Patrick-shaped starfish, sea star. I keep correcting myself because I was always raised saying starfish, but why shouldn't we call it starfish? Take a guess. It's not a fish. So it's not a fish at all. It doesn't fall into the phylum fish do. It's not even a chordata, which is a fish that has a spinal cord. Totally different kind of animal. So it's technically a sea star. You can feel its spikes, how firm it is. The bigger they are, like the softer they are. So you may have gotten a bigger one and a little bit softer, but it still has a spiky skin. On the top of each of these spikes, it has little eyes. Not like that it can see complex things like shapes like we have, but the eyes are for light and dark. So it knows when something's swimming over it. It knows when something's getting close to it, but it doesn't see like we see. It's just the lights are off, the lights are on. That's all it knows. If we flip it over, you can see the rows of feet, and that's where the dark stripe is at. So if I take the probe, they're the little tube feet. They move very easily. These spikes out here don't move as easily, but the tube feet do, all the way down. If you cut an arm off of a starfish, it can actually grow it back. I think they like to be 5-star shaped. They don't like to be 4 because you have to change the name or something. So when we cut this, you're going to pick whichever one you want. I like to pick the one that seems like the oddball or the weakest. This one seems a little weird. Cut it off. Go to about halfway into the arm. Wow, these scissors, I can feel how sharp they are. Spoke too soon. There you go. And you can just get a little pot, put this in there, sprinkle it with some water and dirt, and watch it grow. Not so much. No. There you go. If you look in, you can compare with your picture in your book. So here's the slice we just made. You can see there are 4 tubes. These two, oops. When I cut, I kind of smushed them all. So what I'm gonna do, I'm gonna make another cut, and you can do that too. But you're looking for 4 tubes, so 2 paired tubes, and then there are a bunch of little bumps down here along the bottom called ampullae. And the ampullae are part of the nervous pathway. It doesn't have a brain, but it has nerves into here, so it knows when it touches something. And these little tube feet, when they touch, it knows it, so it knows to suction on or hold on to things. So I'm going to, I'm going to cut right down this pathway again. It's called the ambulacral ridge. Ambula means movement. Lateral actually means tears, but it's a water pathway too. Because those tube feet, they don't work on muscles, they actually work on hydrostatic pressure, which means that water moves them. So to get it to move, it will fill the tubes with water, get it to move, and then relax them. And as they relax, they'll suction onto things. If you ever get a chance, I don't keep plants alive very well. I kill them on accident all the time. But the coolest plant I ever had was called a mimosa plant. And a mimosa plant has a what's the word for it? It's not hydrostatic, it has a hydro system that when you touch the plant, it will curl its leaves up away from you. It's amazing. But what it does is when you touch the plant, it empties all the water vacuoles, all the water tubes, and then basically the leaf goes limp. And then if you leave it alone for a while, it will start firming back up because it refills with water. I knew a guy, this is kind of a weird story, but I knew a guy that was in Vietnam, and they're actually from Vietnam, these plants. And he said it was an eerie feeling because he was in the military, and we were in kind of a war at the time. And he said he would they'd walk through the jungle, and when they saw these plants curled up, they knew somebody was there right before they were there because the plants were curled. He said never knew if it was an animal or if it was another person, so they got like really freaked out by it. So I had to have one of those plants, and of course I killed it. All right, so I just peeled that open. Now we can see everything really well. A starfish has an endoskeleton. It has a skeleton like you, where it's on the inside, which seems really weird because it has an armor on the outside. But you can open it up and you can see all of the ridges, the calcified ridges. It's like a rib cage for you. That's its skeleton. You can see the water vascular system. Depending where you cut, you can kind of see in some of the tube, maybe. You can touch the feet that they connect to and see how soft the feet actually are. And here are the 4 tubes coming out. Here are the top 2 tubes. The top 2 tubes are gonads, so reproductive structures, and it goes in all the arms. The bottom 2 tubes, these darker ones right here, are actually digestive. So these are the pyloric branches or the pyloric system. So that when a starfish eats, it brings food into its mouth right here, digests it, and pushes the nutrients down this pathway to keep the arms all alive. So it's a little bit different than a sponge because it actually has a circulatory like system where it pushes nutrients along. And then you can't really see the ampullae very well. I kind of destroyed them. So sorry. Nope, all gone. Right, so that's one arm. You can see all 4 of those, and they're the ampullae. They kind of just sit right under here, but it looks like I ripped them off when I opened it. So that's one arm. If we go a little bit further in, you can see the mouth. There's the mouth opening where all 5 branches come together. If you look closely, there's a little bump. Oops, I'm kind of peeling it apart. See that little bump right there, that hole? It's a bump with a hole. That's the anus. So its mouth is right here, and its anus is in its mouth. Gross. Yeah. I've been in bathrooms after some people, and I think, oh, how does a starfish do it? Oof, stinky. But that's what it does. Another really gross thing, and if we peel this, kind of bend it back a little bit, you can kind of see the internal organs separating. I want to cut it, but I have a history of breaking and destroying when it cuts. So see this? It's a sac that goes around. That's one of the stomachs. It has 2 stomachs. And this is where it gets really cool, actually. Gross cool. So when a starfish wants to digest something, it can pull the food in and digest it. But its favorite food is a clam, a mussel. So imagine the clam. There's the bottom of the clam on the top. There's the crack. Clams are hard to open. I mean, we have to cook them and then pry them open with a knife, usually, for us to get it open. But a starfish will come along, grab the top and the bottom of the clam, start pulling, take another leg and kind of wedge it in there. And this is the gross part. So now that it has the clam's shell slightly apart, it takes that stomach and spits its stomach into the clam. So the stomach turns inside out, goes into the clam, digests the clam and turns the clam into clam soup, clam chowder, whatever, digests it, and then sucks its stomach and the clam juice back. Pulls the clam juice into the second stomach to digest it and push it off to the arms. So it's kind of cool, weird, cool. But you can, there are YouTube videos out there that you can watch it. Actually, it's slow too. Sometimes watching a starfish do anything, you have to watch it on double speed because it's painfully slow. But yeah, it pushes the stomach out, goes into the clam, and when it goes away from the clam, it's just an empty shell. So one of the benefits of starfish is that starfish kind of clean the ocean bottom too. Clams, when they're overpopulated, destroy the sea plant life and they destroy kind of the ecosystem around them. So clams aren't the best thing. They're not parasites. They're kind of like a, I don't know, like a termite of the sea, I guess. And the same thing with barnacles. So a barnacle looks like that. Starfish can actually eat barnacles off of things because I guess they have crunchy outside and gooey center. So you can see the stomach and you can pull it back and you can look at it. If you want to, you are free to cut it apart. Every time I try to cut it apart, I always break something, but I'll give it a shot. So I'm just going to cut a little piece off here. A little piece. I'm not cutting the whole arm off. I'm just opening it up so that I can see if I can get that part's already done. So I can see more of the stomach. The whole center here, if you look here, you can see kind of a central disc. Everything grows from that central disc. So the entire base out of the center. See right there. So there's your one stomach, the second stomach, and it's poking up. Yeah, that's not. I don't know if you can see the circular pattern all the way around. There it is. I like that picture. And then you have 4 more arms you can play around with, experiment with. I don't know what I'm going to do with. You can go in and you can the stomachs out if you want to. I say experiment. Take advantage of what you've got. That's a weird bump. Pardon me. Next phylum, Mollusca. Clams, squid, octopus, cuttlefish, barnacle. Saw over here. These are all in the Mollusca family. Or sorry, phylum. Very specific. Family is a totally different thing than phylum. So all those are. No, this barnacle doesn't look much like a squid, or a clam doesn't look much like a squid. They have similar characteristics. One characteristic is this mantle, this big body. All through here is the mantle. When you think of a clam has a hard shell on it. The clam shell actually comes from a mantle. So this mantle, even though it feels soft, actually has a cartilage. See the purplish color here? If you feel underneath it, there's a cartilage there. That cartilage is the shell. It's not a hard calcified shell like a clam has, but the cartilage is the same as, and they actually call it a mantle shell. So if I stick my finger on this side, this is the back of the squid. So what's the technical term for the back? Dorsal. So if I flip it over, you can see it's a little bit lighter in color. I can stick my finger in. There is no cartilage here. I can stick my finger all over in there. There's nothing there. This mantle is just a ring. In fact, if you were to catch this without preserving it and you sliced it into rings and fried it, you would have calamari. So if you've chewed calamari, you know they're kind of, well, chewy. And that's what this is, rings of calamari. The coloring you can see, all those little spots, those are called chromatophores. So they're little dots that change colors. An octopus and a squid are both a lot like a chameleon. They can change colors based on their environment. So if they land on kind of a purplish piece of algae, they can match that color to hide. If they are angry, their mood actually changes colors. So this is kind of cool. If they're angry, they turn red. If they're afraid, they turn white. So it's just like us. If you're angry, what color does your face turn? You turn red, right? And then if you're afraid, we get white as a ghost. Same with this. Cuttlefish are supposed to be able to change color so fast that it's almost like a language to other cuttlefish. They use their color to talk to each other. What they say. I don't know who's talking to the cuttlefish to figure that out. All right, so that's the mantle. When we cut it open, if I forget to say it, you should feel all the way along there and feel that cartilage ridge, and maybe even later cut the cartilage ridge, and you can see the cartilage on the inside. So that would be like the shell on a clam. All right, so remember, this side's the dorsal, it's the back, there's the front, there's the eyeball. This, the class of octopus, squid, cuttlefish, they're all called cephalopods. So when you look at, oops, that was in there, yep, there we go. So here are the major classes: gastropods, which means their stomach and their body are the same; bivalves, which would be like a clam because they have two sides; and then cephalopods. Cephalopod means the head. Cephalo, remember, is referring to head. Pod means foot. It means that the feet come off the head. So animals that have their head attached to their feet are called cephalopods: an octopus, a squid, and a cuttlefish. Right, unique. There's the eyeball. I like to look at the lens. If you've had a class with me before, we dissected. Pull the lens out. How do I always, there we go. So the eyes are kind of clouded over. I can actually see the lens right now, right there, as I bend it. I'm gonna grab it. Oh, ew, that was gross. I'm gonna pull that lens out. So this is one of the cool features of actually sharks and cephalopods, is that they have a lens like a human lens. They don't have eye spots that just detect light and dark like a starfish does, or the 100,000 eyes like a crab that we were just talking about earlier. They have an actual lens just like a human, so they can focus long distance or close up just like we do. That's kind of cool. And if you wanted to look, there's the back of the eye. You may actually be able to see some of the muscles that connect the eye so that it can move and turn, and even a optic nerve if you wanted to. As I'm looking at structures, what do we have to list? Arms. So this is a tricky thing. What do you usually call these on an octopus? I mean, I was raised calling them tentacles. That's not true. These are arms. If you grab all of the arms in a bundle and pull like this, see these two that go way beyond? Those are the tentacles. Two tentacles. A tentacle doesn't have the suction feet here on the stem. It only has the suction feet at the tips. So like when you watch SpongeBob, watch Squidward. He actually has hands that are suctiony, but his arms aren't. And they're so accurate. Those SpongeBob people, great anatomists. But they're the two tentacles. The tentacles great, they have a lot of control like we have over our arms, ironically. But they can grab a hold of things like a clam, grab a hold of it, and then use the rest of their arms to pry it open and actually eat the clam from the inside out. They can fight. Some squids can get huge. I can't now, I'm reading how big, but they can compete with whales. Imagine that. Was it 10,000 Leagues Under the Sea? They talked about the giant squids, which a giant squid is not a giant. It's just bigger. And I can't remember what the enormous ones are, but there's some special name for it. But anywho, they can peel with this. These are usually for movement or grip. They don't use these quite the same as the hands. This is like a hand where we have fingers and we know exactly how to use it. These are more like if you had arms with nubs at the tip, so you can grab things or whatever. See how many arms it has. We have two tentacles. Get a tentacle out of the way. That tentacle out of the way. You got 1, 2, 3, 4, 5, 6, 7, 8 arms. So just like an octopus, 8 arms. They probably don't call this an octosquid because it has 8 arms and 2 tentacles. So they should call it a decapus, right? Deca is 10. I think so. It's been a while since I've had to use that term. All right, so we found the eye, found the arms. Beak is also called a radula, R-A-D-U-L-A. And if you're looking at it, again, here's the head, where the eye's at, here's the body. Where would you predict the mouth is at? Usually close to the eyes, right? On most creatures. So it looks like it's this guy right here. That's not it though. This, we'll talk about a little bit more. This is actually a turbo scooter. It's like a super soaker. If you look at jet planes, like military planes, and you see the flame shoot out that propels the jet, that's what this does without the flames. It uses water. We'll be back to this. Look in between all of the arms, right in the center. There's the mouth. The mouth. If we reach in, there's a beak. Feel it? If I tap here, I can hear it tapping. That's the beak, the radula. I don't know if these tweezers, this one's a pretty big one, so I don't know if these tweezers will get it out. Sorry, buddy. Can you see it? That little black beak right there. You feel it. That's it. So if you want to play around with it later, ow, it bit me. It actually kind of did. It has a hook on the tip of it. So it's just like a bird's beak. It has one side that's more of a hook shape and the other one's a little more flat. And if you can get it out, but pictures of it always remind me of, and this is a Star Wars reference, Jabba the Hutt had a little creature that laughed and sat on his shoulder. It reminds me of that thing's beak because it's like dark and light, kind of like lines on it. Now I really want to get it out. If I talk about it, I gotta do it. There we go. Look at that. God, this one is huge. Still pretty fixed, but you can see it better. I think. If I can get it to focus, yeah. Definitely see it better. Now I'm determined. Get the other side. Peel back a little bit more. I don't want to do too much damage because we have, oop, too late. We haven't even cut to the inside yet. Oh well. When these are fresh, they come out easier. Give it up, give it up, give it up. But it also has a tongue on the inside, just like a bird. It's so weird, some of the characteristics that go across species that aren't even related. All right, no, it's not gonna give it up. You can play with it. We've got too many animals to talk about, but that's it, the radula. All right, let's go back to our list. We already talked about chromatophores, so color changers. They can actually be bioluminescent too, so they can glow in the dark. Tentacles, we talked about those. Pen tip. So if you look at the top, sorry, the back, which I'm holding as the top, beside with the chromatophores here, if you come all the way down, there's a pen tip through here. If you flip it over, the siphon is exactly the same place, but on the other side, siphon. If you slide the scissors into the siphon, you can see where it goes. It doesn't go that far. We didn't accidentally cut that. What happens when this swims is it sucks all kinds of water in. This is a big muscle, basically. Sucks water in, pinches this end right here, and then squeezes quickly, and all of that water comes down, goes down through the siphon, and then propels it. 25 miles an hour is the top speed. Siphon, mantle, and then the last thing is the fin. So you have two fins up here at the tip. If it weren't for the fins, this thing would just kind of like spin around and lose some control. It would actually be more like an octopus. But with this, it can swim super fast, faster than an octopus. It could direct its swimming, like wings on a plane. So if it wants to go somewhere, it can fly basically through the water, kind of like a shark's fins. All right, let's check out the inside. Make sure you see the siphon. This is the ventral side. If you don't remember, put your finger in there. If you don't feel anything separating it, you know it's the ventral side, because remember, the mantle is on the back, or the mantle cartilage is on the back. So I'm just gonna go in the middle, take some little bites, snip, 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 snip. I don't want to go too fast because I don't want to cut anything I don't mean to cut, which is good about your scissors. Rounded tip means you're not going to stab an organ, likely not going to stab an organ. Oops. Cut, cut, cut. And you can go almost to the end. You'll feel the resistance. Right about there. Nice. Let me open it up. Squeezy, squeezy, squeezy. I have my pins. So remember, this is like calamari. It's very springy, very chewy. Don't eat it. Pull a pin out. To get it to stay where you want, you need to push the mantle down and put the pin in at an angle. The angle is going towards the body of the squid. So, weird, somebody put them on the end. I never do that. So that would be a way. This is towards, because if you put them this way, that spring will just pull the pins right back out. So we'll go down a little bit, put a second one in. Boop. Come over to this side, put a third one. And yeah, I count out loud because when I'm pulling sharp things out, I always try to remember how many I have. A little bit down, fourth. That angle's not quite enough. There we go. And you have two more pins. If you want, you can pin down the fins or open. So as I look along, just like our internal anatomy, you've got all the organs in a nice little sac, all protected. You can see this clear, no matter how you cut it, you're gonna see it, whether it's here or it's over on this side. It's like a clear saran wrap or cellophane wrap that actually wraps the organs and protects them. So this wrapper is called the peritoneum. You have a peritoneum that goes around your intestines, your stomach. Cut up a little bit. These up here are the gonad, male or female. If it's female, you might actually have it like really full of eggs. Snip. Snip. Over here, you probably see these little things hanging off the side. If you did the shark dissection lab, you probably recognize these. All those lines, as the squid pulls water in and forces it back down, water goes between these little lines. You can kind of separate them or pull them apart. Those are gills. So the water goes across, the gills pull out oxygen, put it into the circulation. You see there's a vessel right here that takes that oxygenated blood up to the hearts. cut far enough. So here's a kidney. I'm going to cut right under the kidney. Boop. 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 Trying not to cut any organs. You can see that reddish colored goo in there. When they preserve these, they push the fluid right into the vascular system. They push it through and they usually push the blood out. Usually, not always. So sometimes the vessels or the heart will burst and you get that fluid leaking inside. So those are the kidneys. There's the central heart right there. Squish, 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 squish. Here's a lateral heart. The other lateral heart's on the other side. The lateral hearts are a little softer. You can actually feel this is a little bit firm from the muscle. So it collects in here, goes to the main heart. The main heart pumps the blood both directions down the squid. So I get nutrients going this way, I get nutrients going that way. If you look at this black tube here, sometimes it's more balled up here at the top and it's a thin tube down here. This is pretty evenly distributed and it's pretty full. But you can see when this was closed, that black stuff leaked through and actually stained the wall here and stained the wall here. It's because that's the ink sac. So if we follow it down, I'm going to go underneath the ink sac. I'm just using the edge of my scissors. I'm not really cutting. I'm just getting underneath that sac. Every organ in your body and every organ in most animals' bodies are protected by sacs. So the ink sac has a wrapper, the intestines have a wrapper, and both of them are wrapped together inside of another wrapper. So what I'm doing is I'm sliding in between those wrappers, just like separating hot dogs, or sausages or something. Oops, accidentally cut it. So the ink sac, you can see the ink is solid because when they put the preservative in it, it becomes gelatinous. Let's see if I can write my initials. Nope. Oh yeah, I can. It's happening. Look at that ink. Ah, takes so much time. Pardon me. So here's how it works. Remember, when they want to scurry away or shoot away, they bring water up in here, they push it down around these organs, not in the organs, and they push it out through this siphon right here. If you look at where the ink sac goes, the ink sac ends right there at the siphon. So what happens is if they get scared and they need to run away really fast, they squeeze their body, they push water this way, and then they pour the ink into the water that's shooting out like a jet. So it mixes the ink with the water at the same time that it leaves, and it creates a cloud real fast. If you take a cup of water and you put 1 drop of food coloring in it, it goes in and it just slowly spreads. But if you squirt the food coloring in with a lot of force, it spreads really fast. That's what the squid does. It squishes really fast right into that water and creates a cloud so they can hide or get away. So if you want, you can actually cut the siphon and see where the end is. Boom. That. There's the inside of the siphon. And there's the end of the ink sac. Just dumps it right in. Goes out. I think if there's anything more significant really to point out. If you want, so this is the intestine. The liver is right down behind it. If you want to see what it ate, remember this thing picks apart like fish with its beak. So you're probably not going to see a whole fish. At best, if it took a big bite, like if you're eating fast and you didn't chew your food well, you might find a little piece of fish eye or something in there, a little piece of fish head. But this, you can feel it. It's dinner. Kind of gross, kind of cool. Cough's catching up. So you can explore all you want through this. You can cut a tentacle and see what it's like on the inside. You can cut the head open if you wanted to. If you want to cut the radula out, you can cut the head, and I don't have it in here to see the brain, but the brain is in between the eyes. So you could always cut that open and check it out. It's up to you. Squid. All right, I'm moving my squid off, so I have 6 pens. 1, 2, 3, 4, 5, 6. Cool, fingers are safe. Let's close that up. Move right along. Crab. Phylum Arthropoda, an arthropod. Here's a fun fact. Not all arthropods live in the ocean. In fact, about half, I was going to say most, but it's actually about half of all of the arthropods live in people's houses. So an arthropod in the ocean, there are crabs and lobsters, crayfish would be an arthropod. Rivers, lakes, streams, whatever around here, you see crawdads, crayfish. Arthropods on land are typically insects, like a cockroach is an arthropod. So when you eat that delicious lobster that you paid 20 bucks a pound for or 30 bucks a pound for, you're eating the water equivalent of a cockroach. So there's your fun fact. Next time you go out to Red Lobster with the family and mom and dad order lobster, say, hey, guess what? That's the cockroach of the sea. It is. It eats all the gunk off the bottom of the ocean. They are bottom dwellers and they love dead things. So what's the term for that? That eats the dead. Necrophagia, eating the dead. Yep. So they're necrophages because they like to eat dead fish that something else killed. They'll just nibble off all the stuff and start eating it. That's what a cockroach does. Just eats all the debris and stuff around, whatever dirty things they can find in a dirty house, I guess. But yeah, arthropods, all arthropods. The defining characteristics, they have segmented bodies. So you can see all in blocks. There's no it's not like us where we uniformly go from our torso down. Oh no, I just popped a glove. They actually have segments. Like when you eat crab, you break different segments of the leg off and they have all these individual joints like insects. They have jointed legs, they have exoskeletons. So what do? Do they have continuously growing or do they molt? They molt. They lose their exoskeleton and they grow a new one. Crawdads do that too. I've only seen it once. I didn't believe it. But if you ever come to an in-house steam when we talk about neuro, I have a fun story about crawdads and molting. I almost spent our time doing it, but not. So, and then they can regrow body parts. So if you break a leg off, this unfortunate guy that has broken some legs off isn't going to regrow them, but it would normally. If it were still alive and it broke that leg off, it would just regrow a new leg. Okay. So things like movement, how it gets its food, how it digests its food. Obviously the movement, how many legs does it have? Well, like I said, mine's unfortunate, but here's where one leg was, 2, 3, 4, and it has this thing up here. That's not a leg, that's a claw or a pincer. So it has 4 pairs of legs, 8 legs. If you look at the different groups in here, crustacean, if you keep going down below, a crustacean will actually go into arachnids too. So it's related to a spider, 8 legs, segmented body, insect. Its shell has use in science. So if we took the shell, it's made of something called chitin. It doesn't matter if it's a cockroach or it's a lobster or a crab. If we ground the shell up into a powder and we actually put it with fats that we eat, it would bind to the fats and you wouldn't be able to absorb the fats. So it was used, may still be used, as a diet pill. They would grind up shells, put them in a diet pill. When somebody ate a fatty meal, they'd eat these shells. They didn't say if it was necessarily crab or lobster. It could have been cockroach shell. They eat it and then it binds to the fat. So they don't gain those calories. Let's see, what are the structure you're looking for? 2 pair of antennae. You should have a long pair and a short pair. It has not been a good day for mine. Oh. There's a short one. Let's see the long ones. There's a short pair. The long ones should have been, it looks like the long one probably came off of here and here. But you have long and short ones. These are called lateral spines. They poke out. Some people call them teeth, but they're not for chewing. Not necessarily teeth. And then the eyes have a stalk. And yeah, it's been a bad day for stalks. So the eye stalk would come out of here, out of that socket area, and the eye could raise up. And Megan shared with me earlier, they have hundreds of little eye sensors on each eye. So they're more like a fly or a spider, not like us. But it's a toad crab, so it's supposed to look more like a shape of a toad. It's like an ugly bug to me, but hey. If you look at the back of it, if there's a line down the center, you have a male. It's horseshoe shaped on the bottom. You get a female. I'm not seeing the line or the horseshoe. Well, there's one other way to find out. Peel this open a little bit. Nope. So inside of here, if it were female and had, oh, no, if it were female and had eggs, it would actually store the egg sac in here. But it is not. When you look at the legs, you can see in this picture, this one, the back set are swimming legs. So this is a blue crab. They have little swimmers. This one doesn't. This one likes to, oops, that's a broken leg. This one doesn't. This likes to hang out on rocks. And you can tell its surroundings around rocks because it looks like a rock and the barnacles hang out. These are more active. That's why they don't usually have barnacles on the back of them. They're very active swimmers. They don't sit still. It's like a boat. If you ride your boat every single day and you leave it in the dock, you don't get barnacles. But if you leave it there for a week and don't move it, barnacles start sticking to it. Same with these. These are not very active, so the barnacles will actually bind to it, and they help because the barnacles will eat some of this bad stuff in the environment around the crab. All right, let's look at the front. Up here, oops. Yeah, oops, that's not what I want. I want this. See, it has, looks like a little piece of armor here, and a little piece of armor here. If you lift it up, it's a maxilliped. It helps with chewing, capturing food, holding food. You see right here on its mouth. It has what looks like a nose. It's not a nose. But it'll take its little pincer, pinch, 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 grab off little pieces of fish or whatever, and start feeding it in here. It'll grind it a little bit with the maxillipeds and then push it into its mouth, so it doesn't have to do a lot of digestion. Once we get inside, we'll look at the digestive organs. Let's go inside. All right, I'm going to go by a leg because the weakest spot in the shell, hard, is by the legs. So if I go, doesn't matter if I go front or back, wiggle it in. I got a little bit in there, clip, I'm through. Boom, I just twisted it. Take another clip. Oops. Oh, actually, I don't have to. Perfect. I might be ripping the intestines apart. So just so I don't do that, even though it wants to peel apart, I'm going to snip around the edges. Right here by the legs, snip, snip. Another little snippy snip. Snippy snip. Now do it to the other side. I'm just basically cutting around the edge of the shell. You can see where the shell, it's like a turtle shell. The top is different than the bottom. And the shell is called a carapace, also a cephalothorax. Cephala means head, thorax means chest. So its head and its chest are the same. I think we got it. Carefully pull open. Keep everything where it was. Not too shabby. Alrighty. Just tilting forward. Oh, looks like some of the intestines are staying up here. So like we saw in the squid, there's a clear plastic. Yeah, crabs is go and get this clear plastic thing at the local store. There's a saran wrap looking membrane that protects all the organs. I'm just trying to go underneath that membrane and peel it off so the organs all stay down on the body and not go up with the shell. It's gonna be hard because I can already see part of them pulled off. All right. Oh, unfortunate. That's okay. And I'll give you a minute or two. I'm seeing that some of you are still cutting, and that's just fine. I know it fits there. It's like it doesn't want to fit there. Okay, so what we're looking at in here, excuse me. First thing you'll notice are these line structures over to the sides. And it's almost like where our chest has lungs on either side. Those are like the lungs, they're the gills. When a crab walks, its legs actually pump its gills. So it's constantly flushing water over the gills to pull out the oxygen. But these are all gills that are breaking off. Oh, this one had a bad day. So mine's kind of, it must have exploded on the inside because all of this rough stuff, there should be a clear line. You should have, excuse me, a spongy little sac here for the stomach. And then right below it should be a heart. It's a little more firm. It looks like when they preserved this one, it blew up on the inside because it's just mush. It mostly looks like coagulated blood. No good, that's no good. So yeah, the heart would be right down there. And it looks like that's what exploded in the stomach. So it pulls the food in. Here's the maxillipeds. You can see there's the mouth underneath, right? You can actually open up the mouth a little bit. It pulls the food in. These glands right here would normally stay up here, and it's the hepatopancreatic gland. So it's a liver and a pancreas. The same organs you have for making chemicals to digest food, the crab has it all in one organ, hepatopancreas. So the food comes in, it's mixed in the first part of the intestine with these different chemicals. It's pushed into the stomach just like we do to store the food and process it completely, and then it excretes it out. And it excretes it out way down here. And this is really weird because the anus is right there. So it comes all the way down the tube, intestines, curls around underneath the shell, up the center, and then boop, it actually poops by its mouth. Kind of odd. Did I vomit or just poop? It's poop. I think I can say things like that around high school kids. I think they get it. Excuse me. And then you can do whatever you want with the spider or spider crab. With the crab, you can explore. You can take the gills apart. You can feel there's a little bit of a protective shell over the gill because it's stuck, but it can peel off. Oh man, that one really stuck to the gill. This was a bad one. You can go and dig down. You can see if you can follow the intestines all the way through. Take the stomach apart. You can explore. You can break the leg off as if you were eating crab legs and look at the tendons. I always think it's fascinating looking at the tendons in a crab. So when I eat crab, I like take it apart slowly and I try and figure out where the muscles are that are controlling each. I'm a total geek. But that's what they are. There's long tendons, not a lot of muscle. It's like your fingers. There's not a lot of muscle in your fingers. What controls most of your finger are actually tendons that are attached either in the palm of your hand or in your forearm that control it. Like if you squeeze right here or push into it and tighten, you can feel the muscles controlling your fingers, especially these. And if you ever want to try this, take your finger like this, put it on a table, and then lift this finger. Put it flat, you can lift it, but then do it like this and try and lift it. It's because the tendons that are coming through, they actually have a common pathway. And when you tighten this one, it makes it so you can't move this one very well, because now this is too loose, the muscles won't move it. It's hard to wiggle. Do the same thing like this. You can just push that. You can't actually move it once your finger's bent like this, because the tendon is so loose, it's being controlled up here in the forearm, that I can wiggle the tip of my finger here, but if I bend it down like this, no more wiggle. But it's amazing how they have such small legs and small muscles, and it's all tendon in there, like a puppet pulling back and forth. So I just get amazed by little things like that. So geeky. Oh, cool. Yeah, we still have time for the lamprey. And then we talk about barnacles on the crab. And barnacles are actually the group Mollusca, which was the squid group. And now for the most disgusting one. So looks like a snake. To me, it doesn't look that much fish-like, but it is actually a fish. In fact, there are two major groups of fish, the bony fish and the cartilage fish. The bony fish have a bony skeleton. They're like salmon, trout. When you think of having fish for dinner and you have to fillet it to get the meat off of the bone, that's a bony fish. A cartilaginous fish would be like a stingray. It has a cartilage skeleton, or a shark. But the lamprey has a cartilage skeleton like a shark, but it doesn't have any characteristics of the bony fish, and not enough of the cartilage fish characteristics, because one important fish characteristic is a jaw. Lamprey doesn't have it. Like we have a hinge here so we can move our jaw. Sharks have it. Sharks were one of the first creatures to actually have a jaw. Sharks have it. Even the stingrays have them. They have little jaws they can open up and go back and forth. But a lamprey doesn't. So it doesn't fit really into either class of fish. So they call this a super species, and there are only a couple of super species, and the lamprey is one of them. So a lamprey, first thing you want to notice is its mouth. I said no jaw. It's just a big wide circle. It's for suction. It sucks on, grabs a hold, digs its teeth into you, and then starts sucking your blood like a vampire. You can kind of see, I get rid of that, kind of see the red and those horn teeth. So I hold it like this, you can stay clear. So the first thing, here are the papilla. These fine little things, those are sensors. Those are like fingers to you. They're extremely sensitive, so that when it attaches to a shark or an octopus or whatever, it feels it, it knows it. It's just like you putting your fingers on something, you know what it is. Like when you hold an apple versus an orange, you know just by touch. It knows, I'm trying to get focused for it. There we go. It knows because of these papilla. Some people call them tentacles. They're not technically tentacles, but oh well. There are three types of teeth. So you have an outer row, the external teeth, and these teeth are actually like hooked. So when they attach to you, they dig under your skin and hold in place. There's a second row here and a third on the tongue, which we'll look at in just a few minutes. But they call this the buccal region. Your mouth is also called the buccal region. Like the muscles that control chewing on the inside, they're called the buccal muscles. So it's a common name. I can see my tongue. You can't see it on the camera, but hopefully you can see yours. It's a nasty little thing in there. And we'll cut it open and look at it. Well, let's get the other features first. So next page. Here are the things we're looking for. So first, we're looking for a dorsal fin. Where would you look for a dorsal fin? On its back, the dorsal side, right? And we talked about this earlier. Here's dorsal fin. Here's dorsal fin, like a sea serpent. So you have the anterior dorsal, posterior dorsal, and this starts with a C, and it refers to tail. It's the caudal fin. There is no cephalofin. If we go all the way back up to the front, we talk about the buccal region. You can see the eye. It's hard to see because it's kind of clouded over, but you see that shape right there? See it? There's the eye. Right there. It's clouded over. See, it has one on the other side, same place. Right in between it, you have a nasal nair. It's like your nose, well, nostril, but it's not for breathing. Water goes in, it stays there, and then flushes back out. It's only a smell organ. So it can smell things inside the water. If it smells a shark, if a shark urinates in the water, it knows. It knows that that's the shark's urine. It knows to go that way because shark has delicious blood. So the urine will go in, activate the brain, and then it gets squished back out. Same with you. When you smell something, if somebody toots in the elevator and you smell it, bad news. That toot went in your nose, activated receptors in the top of your nose, then the mucus in your nose will either pull it forward and you can blow your nose, or you swallow it. Ugh. So next time you step in dog food, think about this. You know you step in dog food because you can smell it and those particles are already in your nose. Right behind it, there's a light colored area. This is actually called the pineal organ. So we have one of these too. It's a pineal gland in our brain that helps us detect light and dark. When it's dark out, our eyes sense less light and the pineal gland starts making melatonin to make us sleepy. When it's light out, our eyes sense the light and shut that off. This lamprey amazingly has the same organ, but it has a window. So it can see light coming down through the ocean, and it knows when it's day and nighttime. So it knows when it's bedtime and when it's time to wake up and chase the sharks. And these aren't just shark bloodsuckers. They'll suck on whales, trout, salmon. A population of these, or a group of these, got released into the Great Lakes in Michigan, and they actually destroyed a lot of the natural fish in Lake Michigan. And I don't know if they completely get rid of the lamprey or if they're still having problems with them, but yeah, they'll destroy any kind of fish. They'll attach onto you. I was watching River Monsters this morning, and the guy stuck one to his neck and was demonstrating and talking about how it felt, and he pulled it, and his skin kept pulling and pulling and pulling and stretching, and then finally it popped off, and he had this big, like, icky spot with a hole right in the middle. Because the tongue here actually has 3 fangs that suck the blood out of you, and we'll get to that in a minute. All right, other features. So we talked about the pineal organ, we talked about the eye, nasal nerve. There are 7 spots along the side. 1, 2, 3, 4, 5, 6, 7. Should have a matching one. 1, 2, 3, 4, 5, 6, 7 over here. Those are the external gill slits. Water goes through, flushes over the gills on the inside, and then comes back out. We are going to cut this guy open. Oh, there's one more feature. Way down here towards the caudal fin, you should look. There should be 2 little tiny holes. They're tiny. There's one right there, and there's one right next to it. In fact, if I squeeze, maybe. Oh, did I just spray the camera? That was gross. That was real gross. Yuck. Ugh. I'm grossed out by it. Anyway, so guess what? That was poop. It just sprayed out. Yeah, the 2 little holes back there, they're called, one's called the cloaca. It's a hole that it can push eggs out of, it can push urine out of, and the other one is the anus. Most fish, like sharks and salmon and everything, have one common cloaca that they do reproduction, urination, defecation, all of that through one hole. Birds have the same thing. So when a bird lays an egg, it lays it from the same hole that it pees and poops out of. But so that's the hole you're looking for. And wow, I should not have pointed that in my head because it came right at me. And I thought for sure it would get the camera. All right, we're going to cut. So we're going to cut first the head in half. Take the scissors. I'm going to hold the left and right sides, and I'm going to cut not a long way, maybe a ½ inch. So about that far. Just so I can cut open the mouth. That's really what I want. And I'm not using the tip of the scissors. I guess these scissors might be sharp enough. Sure they are. I usually use the back part because I can get a better cut with less pressure. And there it is, opened up. I have a cough and I can't make my gross sounds without coughing. Okay, now you can see the tongue. There it is. I have to cover up these words. Actually, I'll just move this. There we go. So there you can see the tongue, and you can feel the horny spikes. Those are the fangs, like a vampire's fangs. You can feel the teeth. The teeth are there just to grip you. They will go around you, and they'll clamp down, but then it sticks its tongue on you and scrapes you and pokes you and sticks the tongue in to try and suck the blood out like a vampire. So if you wanted, you could actually follow the tongue down. You can see the little hole in the center for the pathway. That would go down to the esophagus, which we'll see in just a minute, into the stomachs, into the intestine, and then out that squirt hole that I just squirted. Right, let's go a little deeper. Oh my gosh, totally forgot. Lateral line. It's kind of hard to see on a lamprey. On a shark, you can see it way better. But there's a light gray line, kind of curves over here and goes down the edge. It's a sensory organ. We don't have it, but it's a pressure detector. So if anything gets close to the lamprey, it knows that you're there. You can't get close. When you move water around it, it feels pressure. Just like if I were touching you on the arm, it can feel that water moving here, and it sends it up to his brain, and it helps it localize and know where things are. So even in pitch black water, it knows where its prey's at. That's the lateral line. We talk about tons of that when we dissect a shark. All right, so do I want to do this? No, I don't. I'm just going to go right into the mouth. I'm going to go right by the tongue, and I'm going to clip. Boop, clip. Just clip the buccal region. And I'm just going to open up the belly, the ventral side. If you wanted, you could cut the whole thing in half. And if mom or dad have a chef knife, just you might want to ask them first. But I'm just going to snip at the belly. You might find a couple tight regions, especially right here by the gills, because there's a lot of muscle that help move the gills. Snip, snip, snip. Wiggle in, another snippy snip, snip. I gotta stop saying snip because I'm about to squirt something right in my mouth when I say it. Okay. So I've opened this up. It didn't go all the way down yet. I'm just opening it up so I can see right behind the gill slits, the external ones. Kind of peel this apart. I'm not even cutting it. There we go. Cool. This is probably the best example I've given, because I just peeled the skin off, and you can actually see like blood vessels right here. You can see the row of blood vessels that come down. Get a little closer. There. These red blood bad again. So there's red blood vessels that come down. You can see how they kind of curve off, almost like ribs. This is called the branchial basket. And when it breathes, every time that it squeezes its chest, it pushes blood through there across the gills and off to the rest of the body. So I just peeled the skin, but what you want to do is you want to get a little deeper. So we'll snip it again. So I'm just going to go through and snip that branchial basket. Cut it open, and now I can see all the gills on the inside. You can see the rows of gills. There's a row, there's a row, there's a row. You should have 7 rows that match the 7 external gill slits. Yep. And like I said, if you want, you can go crazy, and after we're done with this, you can cut this in half. You can look and see if you can find the brain, the spinal cord. You could cut it in half like this and look at the brain or the spinal cord, follow it. But we're going to cut and open up the intestines right now, find the heart. It looks like such an evil creature. I don't even know if it has a heart. Right, so I'm just hooking underneath. This is the belly. This whole length of the ventral side is the belly. So I'm just hooking under here and snipping a little bit. So I try not to cut anything I don't want to cut. I definitely don't want to squeeze the end, even though I probably squeezed everything out of it already. Now I can open it. Ooh, this guy's muscular. A lot of muscle. Excuse me. Hepatopancreatic gland. So it's the liver and pancreas. So it sucks blood, goes down its esophagus, goes into the stomach, which would be right up in under the gills, but behind the heart. Here's the heart. I move away. So esophagus to stomach, and then that's where the hepatopancreatic duct is. That pushes enzymes to help digest the blood. As it's pushing it, it goes down, down, down the intestines, all the way down, all the way down, all the way down, all the way down. Keep it going all the way. Oops, easy to find. And it'll go all the way to where it's squirted. I'll keep my mouth shut when I do this. Actually click underneath, pull it up. Keep going, keep going, keep going. Ooh, keep going. Oh no. It's right about there. And it keeps going until right there, the anus. So the first hole. Second hole, reproductive. See the reproductive organs that go up, curl up here. Pardon me. And now I want to go back for that heart. So I'm going to go back up here, the gills. The heart is right below the gills. There it is. Got you. There's that liver pancreas. Here are the gills. If you can see it, come up closer. And there's the heart right in between. Ooh, I kind of shredded my heart. That's no good. But you can see the connections as I'm pulling it out. You can see the blood vessel that goes right up into the gills. You can see the blood vessel that goes down to the rest of the body. There it is. Boom. That little guy. Isn't it amazing? That little heart circulates for the entire body of this. Amazing. Yeah, you have a minute. So another thing you can do, like I said, you can cut up along here. The brain is in between the two eyes. Or I'm going to cut right here. I want to see the cross section. This is actually a respiratory pocket. Helps it to float a little bit. There you can see a blood vessel. Oops. So there's an aorta, which carries blood away from the heart. There's, looks like it right there. Yeah, there's a vein. Veins aren't strong. Aortas are strong, so I can push it shut and it pops back open. But a vein, you just have to feel for it and you'll find the tube. And then right here is what they call a notochord, which is actually the spinal cord. And you can feel the cartilage that protects just like our vertebra. These are chordata, which means they have a spinal cord just like we do. And then there's the dorsal artery, another blood vessel up there. You can see the tubes are carrying the blood all over. This is all muscle, so they can slither through the water. Oops, I'm sorry. There we go. Weird, every time I get close, it goes away. So artery, vein, cord, and then another artery right up here. These are all pieces of muscle. There's the abdomen. There was the liver, pancreas, part of the gonads, intestines. You can follow it all the way down. So you can slice wherever you want. You can see the cord's going to stay the same. Some of the structure will change as you get further down. Ooh, a lot of dissecting. Cool. So you can do whatever you want with this, but my last job is I'm going to show you how to clean it all up. The first thing you do, take your pins and put them in sideways. Don't put them like this because if you squeeze it, they're longer than the pad is thick. So you put them like this, fingers are always safe. And you, like I said, if you want to keep dissecting, you are welcome to. It's kind of fun to find things. So sideways, and then I'll put this at an angle. Whoops. So it doesn't poke me. I'm going to put my squid right over it. Boop. And you can cut the squid's head open and look for the brain too. Crab. I'll keep the sponge. Put the starfish pieces, crab legs. Yum, yum. If you want to keep the book, it's up to you. It's probably stinky nasty, but What is this? Not important to me. Okay, scissors. Everything's piled in one thing. This is how you pack it all up. The side that's the bottom is over here. When we laid it all out, we made sure we knew what that was. So that means this is the top of the bag. The hand that matches the top of the bag, I'm ungloving. Putting my hand inside the bag. So I can touch all of this with my gloved hand, but my clean hand's going in like this. And I'm just rolling the bag over it. So my glove only touches the dirty side. Like that. And I have it most of the way. Take the glove off, throw it in. Everything out here is clean, so I just like a magic torch. Contain. Snatch the air out of it, roll it a little bit. Ooh, I felt the crunchy spider in there. Spider crab. And then if you have your original plastic bag, you can throw this in. Otherwise, tie a knot so things don't fall out. That's everything. You can put, if you have the original plastic bag that was clean, you put it in there. Otherwise, joke it. So thanks for playing along. Thanks for cutting things up. Thanks for getting gross. I love this. If you get a chance to do the shark or the internal organs ones with like brains and eyeballs and stuff, definitely do it. Otherwise, hopefully I'll see all you back here at one of our real stems when we open our doors again. Hopefully soon. So, have a good weekend. Thanks for playing.